Thank you for the introduction. I first of all like to thank the organizers for inviting me to this London calling. This is very exciting, and it's a great honor and pleasure for me to talk about recent efforts on making a comprehensive atlas of human RNA structures and their modifications. Yeah, this is a brief overview of my talk today. The main point or topic of my talk today is the full-length RNA transcriptome analysis. So mostly I want to talk about the unpublished data generated by Kazuhiro in my group, and this is all the data generated by Oxford Nanopore sequencing technologies. So I have two sections in my talk. The first, I want to talk about the data generated by cDNA sequencing, and then I move on to data direct RNA sequencing. So the first, I want to talk about our de novo human transcriptome assembly using full-length cDNA sequencing. And this revealed, very surprisingly, many new novel gene candidates in human that are expressed in a very cell-type specific manner. And just speaking about human transcriptome is highly complex with many unknown molecules. The reason for this is RNA expression is highly dynamic across cell types and states, but full picture still remains incomplete. This is the example of GenCode gene model. So shown in blue, the protein coding genes, and even from the one single gene, there are many different isoforms expressed, and also there are also many non-coding RNA genes depicted in green. These are non-coding RNA that can function potentially as RNA on their own without being translated into proteins. But they're really, really diverse molecules expressed in human bodies, and even new genes or isoforms can be expressed. And in our group, we are studying, we're trying to understand gene structure and regulation to understand what is written in our genome. And upon activation of transcriptional mechanisms like enhancers and promoters by transcription factors binding, and then RNAs transcribe from genes. And at the very beginning of transcript, which are 5' end of transcripts, so the 5' end is capped, and then splicing removes introns co-transcriptionally, and at the 3' end, polyadenylation happens. So the mature form of RNAs looks like this. So it has 5' cap, and then at the 3' end, it has a poly-A tail. So to understand genes, we're trying to sequence the entire stretches of RNA molecules. And as we all know, the conventional short-read RNA sequencing provides fragmented information about the transcripts. So this can sometimes puzzles the analysis of the transcriptomes. But the long-read sequencing is a powerful approach where you can identify transcription start site and also splice sites and also transcription end site. However, in reality, there are many complications. So the long-read sequencing may not always capture the full-length sequence of RNAs. So what I mean by this is we typically use oligo-DT to initiate reverse transcription. And if the RT reaction goes all the way to 5' end like this is very good. But what happens also is there are many internal A-rich regions to which oligo-DT primer can hybridize to, and then so this is called internal priming. So this can result in truncated cDNA. Also, RT reactions, reverse transcriptions, do not necessarily go all the way to 5', and this happens oftentimes at the GC-rich region of the transcripts. So we want to only take this full-length cDNA. So the preparation of cDNA is also a very critical step to do a long full-length analysis, and this is actually the key point of what we're doing. So we're working on this method called, we named as cap-to-tail sequencing. So this is to do a full-length RNA sequencing analysis. So basically, the protocol, we use this PacBio PCB111 kit, and we basically start the same way. The protocol starts with ligating overhang adapters, and then we use an adapter-specific primer, including that are designed in such a way that poly-A tail is also included in the cDNA. And when we do RT reverse transcription, we use a higher temperature, starting with 42 degrees, but then gradually increase to 60 degrees to destroy the secondary structure that oftentimes happens at the GC-rich regions. And then next, we use GC-tolerant PCR enzymes to ensure that PCR amplification happens equally even to GC-rich regions. And then followed by cDNA site selection, and then sequencing using Oxford Nanopore flow cell. So the key 2 points is reverse transcription at higher temperature to dissolve secondary structure, and also GC-tolerant PCR enzymes. And this is what we could get by improving this cDNA preparation. We could make cDNA. So this is the distribution of the mapped reads. So this is a read, not the isoform that we called. And then you see this average read length of 2,568 base pairs strong in K562 human cells. This is almost matching the median length of mRNA transcripts. So this allows a very comprehensive and broad analysis of the human transcriptomes. And on top of this, we did additional computational filtering to ensure that each read is full length. As I said earlier, the Nanopore data contains poly-A tail. This is a very nice, because you can see that this is the 3' end. So we extracted reads that only have the poly-A tail, removing reads that do not contain poly-A tail. And then on top of this, we performed cap analysis of gene expression. This is a method to sequence a 5' end of transcript. So it's a short-read sequencing method to look at the transcription peak site. And we extracted, again, only reads that have the overlap with the cage peak. So this is to ensure that each read is supported by cage 5' and also the poly-A tail at the 3'. So this is a read-level full-length transcriptome analysis. And we called isoforms if each isoform is supported by 3 or more reads. And this we performed not only just on cell types, but as you see here, we performed this method to almost 500 different human cell lines and primary tissues and primary cells. And as you see here, we collected high-quality RNA samples. If you look at this, the read values, RNA integrity number, so this is almost 10. And so this diverse and high-quality RNA samples for comprehensive analysis is performed. And as you see here, we generated data in a high-throughput manner using Oxford Nanopore technologies. The low input requirement for Nanopore sequencing plus the fast protocol actually allowed us to do this in a high-throughput manner. And also, we together performed cage short-read RNA sequencing, and also Illumina short-read sequencing, and also attack-seq protocols to ensure that this is promoter. So all this systematic large-scale data set was generated to systematically characterize human gene structures. And this is what we could get using our data sets. Based on this 560 million CT-seq reads from almost 500 human samples, we detected isoforms, and we compared the obtained isoform with GenCode 47 transcript models. And we could see that there are a lot of altogether, we could detect 131,720 isoforms detected from our data set. And as you see here, half of the isoforms that we could detect matched with the GenCode version 47. But surprisingly, we could identify another half of transcripts, isoforms, that were not included in the GenCode version 47. But most surprisingly, there are around 2,500 novel gene candidates, giving rise to 3,557 novel RNA isoforms. And this is one example of a novel unannotated human gene. So this novel gene candidate is located on the antisense strand of a known gene. So you see here, this is the intronic regions, and then this is the three exons, and sometimes 4. For one read, it has 4. So it's supported by, and then each read is supported by the presence of poly-A tail, and then epigenetic modifications also support the presence of this gene in the antisense strand of a known gene. And looking at the expression pattern of these novel genes, so this is what is shown here is the expression pattern of known genes. So each dot is gene, and then the expression levels of median expression across 482 samples were detected on the x-axis compared to the maximum expression across 482 samples were plotted on the y-axis. So if the plot is close to diagonal, it means that it's ubiquitously expressed. But if it's expressed close to the y-axis, it means it's cell-type specific expression. And as you see here, like albumin, which is liver-specific, is plotted close to y-axis. And as you see here, the most of novel gene candidates, these are expressed in a highly cell-type specific manner. And these are maybe one of the reasons why these 2,500 genes were overlooked previously. And also the next question is, yes, we identified thousands of new genes, and the question is whether these are translated or not translated. And actually, the answers I don't know whether these are expressed, but we integrated our data sets with proteomics, and this is just one example. So this is a novel gene candidate expressed in K562 cells, and the predicted open reading frame is present, and then this peptide was detected in mass spectrometry analysis. But this happens not all of them. So I believe that a large fraction of them are non-coding, but they are also a good fraction of potential protein coding genes expressed. And the thing that we want to do is also to look at things that have never been looked even before. So what we want to do is, yes, the CT-seq protocol generated reads that are longer than what has been achieved before, but we also want to look at ultra-long. Here, in this case, it's a sort of site selection using blue pippin to collect for very long cDNA selection. And then we could achieve this, the ultra-long CT-seq data, and the median length you see here is about 7 to 8 kb. And this data actually included isoforms expressed from BRCA2 gene. So BRCA2 is a gene involved in breast cancer and also ovarian cancers. And as you see here, that just a conventional CT-seq protocol did not detect this expression, but this ultra-long CT-seq data detected many isoforms expressed from BRCA2. And interestingly, there are many alternative splicing events from the BRCA2, but we could also identify some novel exons that are not included in the database. So this also indicates the importance of getting actual data from the samples that you're studying. That helps us to reveal many novel expression patterns. So just a brief summary for this first part, which is leveraging nanopore sequencing and a diverse collection of high-quality human samples. We systematically identified novel human genes, and I believe the function analysis is the next frontier. And then I'd like to move on to single-molecule native RNA sequencing at scale, and this revealed the complexity and modification in human transcriptome. And this is the kit also released from Oxford Nanopore. And this is a really amazing technology by which you can directly sequence RNA so that you can also, the data includes also RNA modification data. So the protocol, we used 2 to 5 microgram of RNA, and then attaching RT adapter to perform RT, and then additional attachment of the sequencing adapters. So the protocol is very short, like almost half a day before loading onto the flow cell, and this is very exciting. And we actually did a little bit of additional stringent filtering, purification of longer size RNAs so that we can compare data sets we generated from cDNA-seq and then direct RNA sequencing. So this actually allowed us to do this direct RNA sequencing with a median length of over 2 kb and about 50%, or in this case, 47% of raw data, raw reads, were supported by CAGE, meaning they are full-length reads. Yeah, and we are also very happy to see this very nice reproducibility between the replicates. You have here the replicate 1 comparing to the expression levels achieved by replicate 2. So this is a high correlation, the Spearman's correlation of 0.955. So this is also indicates a good quantitativeness we can achieve using direct RNA sequencing. Also, we compared data we could get from direct RNA sequencing versus what we could get with CT-seq protocol. And although there are some transcripts detected more or less preferentially in either methods, generally we could see a good correlation. Yeah, and then we performed direct RNA nanopore sequencing on the diverse human samples. And as you see here, the RIN values, the quality of RNAs are indicated here. It's almost 10 or something higher, or close to 10. And then we made like a 3 million reads, and on average, like a 1.2 million reads were obtained for each library. But very interestingly, also, so this is showing the distribution of the median length and also N50, and so it's all more than 2 kb. So the long-read sequencing analysis can be achieved using a direct RNA sequencing. And the beauty of this method is that the data actually contains RNA modification data, so we could identify numerous regions of transcripts that are present with RNA modifications. So this is looking at M6A sites and M5C sites, and also pseudouridine. As you see here, so some reads, some regions are supported by many reads, and sometimes higher, almost close to 100 or 90% of reads contain specific RNA modifications, or there are also some cases where only 10 to 20% of regions are modified with RNA modifications. And globally, from close to 30 samples that we did for this analysis, we could see that some RNA modification events are very highly cell-type-specific, and some regions are ubiquitously happening. And this is one example where these RNA modifications that you see here, so these are present in K562 cells, but not in HCT116 cells. So cell-type specific RNA modifications shown together with the full-length isoforms. So this is, I believe, the first event we are starting to look at these exciting data. Yeah, so the disruptive innovation of this direct RNA sequencing has made it possible to detect RNA-based modifications, allowing comprehensive analysis of both RNA structure and its chemical modifications. And this is very exciting. And our rich data set of the human transcriptome encompassing both cDNA and direct RNA sequencing across diverse cells and tissue types offers an unprecedented opportunity to explore transcript diversity. And Oxford Nanopore sequencing uniquely enables this depth of insight, capturing full-length RNA isoforms, including those from even new unannotated loci, as well as RNA modifications, and providing a powerful lens into the true complexity of human biology. And lastly, I want to acknowledge all the persons who contributed to this project, and I'd like to give special thanks to Kazuhiro, who is also sitting here, who made these large data sets, and also he very nicely prepared the high-quality RNA samples, which is a very critical point to make good data. Yeah. Oh, it's not. Yep. Yeah. So with this, I'd like to conclude, and thank you for your attention. And I'm happy to take any questions. Thank you very much.